Okay, before we get started, I have a couple of announcements. I'll try and do at least one of these announcements every week, and that is the middle screen up here. This is the Lester Newman seminar series in the biology department. Lester Newman was a professor of genetics here starting in the mid-60s and just retired a couple of years ago. He just gave $100,000 to the department for supporting undergraduate research. So keep your eyes out for support for undergraduate research in the near future. This week's seminar is Adapt to People or Go Extinct. Sounds like an interesting topic. How spotted hyenas survive while populations of other large carnivores decline. This will be in room 247 in SRTC, which is literally straight across the hall from the biology office. Usually these seminars are a lot of fun. There's free food, good deal, and coffee, even better. But make sure you get there early so that you can get the food and coffee. Highly recommended as far as that's concerned. So that's the first of the announcements. The second of the announcements is a flyer that I got from OHSU, the National Primate Research Center. Sorry. Pardon? Oh, the timing. Yeah, it's at noon. Noon to 1. Yeah, so if you want to get there early, to make sure you get the snacks and coffee. A little before noon is a good idea. I don't know if there's another class in there beforehand. Yeah, so OHSU, their Primate Research Center, which is at the West Campus of OHSU, does these really cool science cafes. Basically, low-key discussion about some really current and active research that's going on there. The first of them is February 6th. Nancy Hagewood, who's done some really amazing work on HIV vaccine development. And again, they do some really, really nice stuff there, and they're great conversations to have. It's out at PCC Willow Creek. A little bit of a trek, but definitely worthwhile to go and check some of those things out. The other announcement that I had, those of you who've been looking carefully at the syllabus may have noticed, man, this is almost impossible to read. Well, be that as it may, it doesn't really matter. That there are a couple of lecture dates that are in italics. I'll be over just a sec. And one of those is because you may or may not be able to see this. Again, you probably can't read here. This is the University of Florida, where I'm giving a talk on February 8th, which makes it tough for me to be here on February 9th. So we'll have a guest lecture. That'll be Dr. Mike Bartlett from the biology department, who will be speaking then. Your question? Yes, I will post that flyer on D2L, exactly. And I've been posting a couple of things. I've been getting some internship things, and also there was a job recruiting thing. So I've been posting a couple of things on D2L there, usually in the discussions section of D2L. So other questions, comments, worries about where we are yet? The other thing which is being posted on D2L are the clickers. I have not updated them since I did so on Monday after class. So if you've registered between now and then your clicker, that may not be registered yet. But just to check, this doesn't count yet. I want to see how people are doing with clickers so far, so go ahead and pull them out. This is our second clicker question. And again, this doesn't count. I'm not going to start counting until next week. And of course, that's a week from today because Monday is off. So this actually works now? Yes, except that it's a little bit off. It's not telling you how much time there's left. There's the most molecular diversity in which of the following? Archaea, animals, plants, fungi, or humans? Doesn't seem to be registering, which is, oh, this whole thing here. Close. Okay, now it's going. There we go. I need to turn off that default. And again, feel free to talk to your neighbors about this. This is supposed to be interactive. And it's your last vote that counts. So vote early, vote often, only one of them counts. At least in this class. 5. Okay, 94 people have clickers here now. That's good. Okay. And most of you think archaea, which is good because that's what I think too. If you think about the large groups of organisms, it really is, if I'd put archaea, bacteria, and eukarya up there, then it would have been a way harder question. But animals, plants, fungi, and humans have ridiculously small amounts of molecular diversity relative to archaea, bacteria, or eukarya. So I'll also select the answer here. This, again, should show up this afternoon in D2L, and this way you can check and make sure that you're appropriately registered. Okay, I wanted to continue talk about a little bit about, beyond going to genomes, a little bit about where new genes come from, how genes are invented, etc. Because given that tree-like structure, again, with archaea, bacteria, and eukarya, presumably everything came from a single common ancestor. At least all extant cellular life came from this single common ancestor. But there are lots of different genes in different genomes. And so how do you go from that one set of genes in that ancestral cellular organism to a whole bunch of other ones? So there are a couple of different ways that this can happen. One is sort of the obvious one, mutation, just a single change in a single gene. And I brought my DNA model here. There are all kinds of problems with this DNA model, which is a great review in thinking about DNA. And I'll try and get down off the stage and talk about that a little bit later on. But just, single point mutation that'll generate some kind of diversity. The next kind of thing that can happen, second one down here, is very, very frequent. This is the process of gene duplication. You go from one copy of a gene to 2 copies of the gene. Usually this has to do with cell division processes, and cell division has just gone slightly wrong somehow. Gene duplication of itself is probably not that interesting, but the really neat thing about gene duplication is you have 2 copies of one gene now. If one of those genes is absolutely essential, you can fiddle around with the other gene, or I should say evolution can fiddle around with that other gene and come up with some new kinds of functions. Or, as you have on the third line here, is have mixing and matching between those 2 different genes. And we'll talk about domains in terms of protein structures a little bit later on, as far as how you can generate these new kinds of genes. And then at the bottom is my favorite version of how you get new genes, because viruses rule, obviously. And that's the process of what's called horizontal gene transfer. And it's basically a gene from one organism getting moved to another organism that's not directly related to it. And that process clearly can generate a whole lot of diversity very, very quickly. Because, well, hopefully here, I'm sorry for those who are red-green colorblind. My brother's red-green colorblind, so it's harder to see these things. But you can hopefully see that there's a red gene from this sort of red, yellow, orange genome that's now ended up in this greenish organism over here. And clearly you can introduce a huge amount of diversity through this kind of horizontal gene transfer process. And we'll talk about how that works. Again, a lot of it has to do with viruses a little bit later on. But first I wanted to mention a very, very important point. And we talked about this a little bit last time, sort of conservation versus diversity in terms of thinking about how to make the kinds of trees that we talked about, how people know that archaea are different from bacteria and eukarya. We need a terminology to talk about these kinds of things. And so the overall sort of terminology I like to think of as something called homology. So all that homology means is if you've got 2 sequences, they are evolutionarily related to each other. And how do you know that? Usually, it's because you put one sequence into a computer and hit search, and you found another sequence, which is similar to it. And that's how a huge amount of biology is done these days, is you give your favorite gene, your newest gene that you found, you compare it to everything else which is out there, and if you can find a homologous gene, presumably related by common ancestry, it'll hopefully tell you something about that gene instead of having to do all of the time-consuming experiments to figure out what it actually does. There are two kinds of homologs, the so-called orthologs and paralogs. And this is a issue that often comes up and people can sometimes get confused by it. I got confused by it the first probably 20 times that I went through it. So there are two different ways of looking at this. Basically, it has to do with whether the gene has arisen from a gene duplication in a single organism, or it's arisen because that single gene has been in two different organisms and then is modified in each of those different organisms. So I actually like to start over here on the right-hand side. If you have a gene duplication and then one of those genes changes, it's a gene duplication, so it's clearly homologous. It's related, has a common ancestry, but it's in the same organism. So these are now paralogous genes. And as I mentioned before, with the gene duplication, if one of these genes is essential, say this orange one, the yellow one can be free to mutate, change, potentially get some kind of new activity. On the other hand, orthologs, and again, you have a sequence of gene B, you compare it to things in the database, and you find that there's a homolog in a different species that seems to have come from a common ancestor. These are now orthologs relative to each other. So orthologs, paralogs gene duplication, orthologs due to speciation in different species. And in this case, what do you think would be the case if this is an essential gene? Are you going to be able to mutate these genes a whole bunch? No, probably not, because it's going to still have that essential function in that particular organism. Now, this is the absolutely oversimplified version of looking at it. This process happens in all organisms all the time throughout evolution. So you end up with a huge mixture of these kinds of orthologs and paralogs. And in these homologous genes, what you end up with are things called gene families. So these are homologous genes that are found in lots of different organisms, and often these would be what? Orthologs or paralogs? Lots of different organisms? Those are orthologs, but also often lots of paralogs because they're gene duplications that have happened in a single organism. And this is just another way of looking at that here. Here we have a specific example. This is tubulin. There was a gene duplication and divergence that happened, generating alpha tubulin and beta tubulin. These again are what? Orthologs or paralogs? Paralogs, exactly. And then speciation happens, and you have one organism with alpha and beta tubulin, and a different organism with alpha and beta tubulin. These are now what? These are going to be orthologs relative to paralogs. And you can do sequence analysis, again, comparing sequences to each other. And what you'll find here is that all of the alpha tubulins, and again, this is just comparing the sequences, counting the differences. Things are closer together, they're more similar in sequence, further apart, more different in sequence. That all of these orthologs, the alpha tubulins, are more similar to each other than the beta tubulins, but then they also have all these paralogs. So again, here at the top, again, I should put my glasses on so I can read these. Humans, flies, worms, and yeast, etc. So people clear orthologs, paralogs? When somebody asks you questions on these, yes? Mm-hmm. So the question here, I'm just going to repeat this so it'll be recorded. The question is, when you have gene duplication, does one of those genes go inactive? And the answer is sometimes yes, sometimes no. But the big advantage of the gene duplication is that you can then modify that second gene if the first one is essential, and that could generate something with new activity. Most of the time, and we'll talk about this a lot more when we talk about mutations and things, most of the time when you make a change in a gene, it's actually deleterious. So yeah, often it will be a loss of function. So that's what's happening with this gene duplication and changes. Again, usually these will be single mutations or maybe rearrangements that happen in one particular cell. Then horizontal or lateral gene transfer. This is how you get a bunch of new genes. And of course, it's the most interesting because it has to do with viruses. Basically, viruses I like to think of as horizontal gene transfer machines. They take nucleic acid and put it into a new cell. Usually it's the viral nucleic acid, but very often viruses will pick up some nucleic acid from the first cell that they've infected and put it into another cell that they've infect after that. And that's not necessarily going to be one which is related due to a linear mother to daughter kind of lineage. So bacterial viruses in particular, but viruses in general, are really good at moving genes around. So a lot of horizontal gene transfer has to do with the movement of viruses. Definitely true in single-celled organisms, particularly the bacteria and archaea. And the nicest example of that, because E. coli has been really well studied, is if you just look at different E. colis, up to 20% of the genes in each E. coli seems to have come from a different place, so not linearly related. So huge amounts of horizontal gene transfer have happened in many bacteria and archaea. Probably the origin of life, back at that root of that tree, there was a huge amount of horizontal gene transfer that was going on there as well, viral or other mechanisms. Our, I guess, favorite way of thinking about horizontal gene transfer, you think about haploid and diploid organisms, like us. What do we do then? We take nucleic acid and genes from two different organisms, and we mix and match them. That's also a way of thinking about lateral gene transfer, because the genes in mom versus the genes in dad have different histories. When you go through meiosis, haploids then becoming diploids, that's also a mechanism of horizontal gene transfer and getting more diversity into your population, into your organisms. So why do I spend so much time talking about this? Well, it turns out that there are, again, through this duplication process and probably also horizontal gene transfer, generations of lots of different gene families that are found in all kinds of organisms, or at least most kinds of organisms. And it's up to 60-odd gene families, particularly present in the translation apparatus, which is exactly what you'd expect. Again, when we talked about that big tree, bacteria, archaea, and eukarya, originally shown from the ribosomal sequences. And the ribosome has been conserved for all cellular life that we know of, at least to this point. There are a number of other things usually involved in pretty central processes, either in energy metabolism or in replication or transcription. Replication, again, DNA to DNA, and transcription from DNA to RNA. So we've got all these gene families, got all these wonderful sequences. How do we figure out what a gene does? And this is the hard part. Doing sequencing, and we'll talk about this right at the end of the term, is really, really easy, and it's getting really, really cheap. But just the sequence doesn't really tell you very much. So there are a couple of different ways of trying to figure out what, and I'll talk about this many times in this class, YFG, your favorite gene, does. One of those is genetics. And how many, again, you people told me last time, how many people taking genetics now? Okay. What do geneticists do? What does it say up here? They break stuff and try and figure out what they broke. Which, as certain experimental geneticists, that's really what happens. You have a mutant, and then you try and figure out what the phenotype is of that mutant, which will tell you something about that particular change in that particular gene. And this is great if you have a genetically tractable organism. And unfortunately, that's not true for many, many, many different organisms, and also doing experimental genetics on organisms that we care about, like whoever's sitting next to you, doing that experimental genetics is ethically very problematic. So that's one way of trying to figure out what a gene does. Another way is biochemistry, which is kind of the exact flip side of genetics. Genetics is looking at what a gene is doing in the context of an organism. Biochemistry, you take one gene, you figure out what the product is of that gene, and in isolation, what does it do? And this is incredibly powerful, and we've got all kinds of wonderful techniques. Again, we'll talk about those towards the end of the class, where you can literally take one gene, make the protein from it or the RNA from it, and then figure out exactly what it does, exactly what the high-resolution structure, and structure tells us function, of that one particular gene. But how it works in context, you're not going to get necessarily from the biochemistry. You needed some genetics as well. So it's really the combination of biochemistry and genetics that gives us a really good handle on gene function. And in this day and age, as I mentioned when we talked about the gene duplication here, what a lot of people end up doing is so-called bioinformatics. This is a, again, ridiculous term. There's a class going on this term on bioinformatics, which is also nowhere near enough time to do all of bioinformatics. But when people talk about bioinformatics, usually it's talking about doing sequence searches and comparing what you've just found, your favorite gene, to anything that anybody else has found. And if you're lucky, somebody's done genetics and or biochemistry on that particular gene, and it'll tell you at least where to start the rest of your experiments in terms of trying to figure out what else is going on. So that being said, and you're all happy with orthologs and paralogs, right? So you can answer a question on them. Good. So start. Which of the following is the best description of genes that are in a gene family? They're homologs, they're paralogs, they're orthologs, they're paradoxes, they have identical sequences. D. Again, feel free to discuss this. This is sort of the point. It should be loud when I give these clicker questions. Ten. 3. Okay, most people think homologs. I also agree with you. Paralogs and orthologs, those end up in the gene family themselves. So when I talked about those gene families on the table, which I probably didn't do a great job of discussing, those were gene families that are found all the way across all cellular life. So all those ribosomal sequences, all those ribosomal proteins that are in families, you find them in all kinds of different organisms. You find them in different organisms, what do they have to be? Orthologs. If you got gene duplications in one organism, those are the paralogs. So homologs is sort of the higher order process that we're talking about there. So that is correct. So with all of these gene families, what do we end up with? Genomes. Genomes have all kinds of different sizes. We talked about the E. coli genome last time. That's a sort of medium-sized bacterial genome. We also talked about the mycobacterial genome, which I think is right here, under 1 million base pairs. Again, E. coli is about 4 to 5. Again, depends on which strain you're talking about, how much lateral gene transfer has happened in that particular strain of E. coli. And then us, we're up here, about 3, and this is the haploid genome now, 3 billion base pairs for the human genome. Yeah, in the back.
The last class you said, can you define the genome again? The last class you said was all the DNA in the organism, or there was a discrepancy in that?
Okay, yes. So, I'll paraphrase your question and tell me if I'm getting it right. What is a genome? How do you define a genome? So a lot of people will define genomes, and I say a lot of people, that doesn't include me, well, just the genes that are in any given genome. I think that's an underestimation because there's all kinds of other sequences, and that's partly what this slide is about too. There are regulatory sequences, there's a lot of quote-unquote junk DNA, which we're figuring out is probably not as junky as we thought it was in the first place. So I would say when we're thinking genomes, it's really the complete nucleotide sequence of a particular organism. One thing that we didn't talk about, and I wasn't really planning on talking about. The book gets into this, but who cares about eukaryotes? You've got mitochondrial genomes, you've got chloroplast genomes as well, if you're talking about plants. So is the genome of an organism, does it include the mitochondria and the chloroplasts, et cetera? So people will argue a little bit about exactly what constitutes the genome. As far as I'm concerned, it'll be the nuclear genome or the whole genome if you have an anucleate cell like a bacteria or an archaea. Okay, there's sort of questions on the genomes. But yeah, so there's a huge variety in genome size. And the bottom down here is a log scale in terms of numbers of nucleotides. And so some people argue the humans were higher organisms. Lots of my friends in graduate school used to work with Saccharomyces cerevisiae. Yeast just said, we're just bigger eukaryotes, we're not higher eukaryotes, and none of this higher and lower stuff. And size of genome. Size, in this case, does not matter, whatever anyone will tell you. Size doesn't matter of genomes. If you look at some of these protozoal genomes, amoebal genomes are literally 2 orders of magnitude bigger than the human genome, which means they're even higher organisms, right? Because they've got bigger genomes. Well, what makes that difference in terms of the genomes? Most of it is repeated sequence. And we'll talk about some of the repeated sequence in the human genome. A lot of that's viral sequence, actually, too, which is really fascinating. One of the reasons that viruses are so cool. But lots and lots of repeated sequence, but also a lot of that regulatory sequence. So getting back to your question about genes versus genomes, it's not just the collection of genes, it's also all the extra sequences which are there. And so a lot of those extra sequences, you seem to have a lot more of those extra regulatory sequences in some of these organisms that have larger genomes, like humans, relative to some of the bacterial genomes. But also, if you look at some of these other eukaryotic genomes, there's this one right here, yeast, which is actually a pretty small genome. It's only about 13 million base pairs in length, so not much bigger than E. coli. And having a smaller genome, less of repeated and extra DNA, means it's actually considerably easier to study, particularly from a genetic point of view. Because if you're changing genes, fewer genes, smaller amount of DNA to work with, those are often really good organisms. So another thing to think about in terms of the human genome, 98.5% of this says non-protein coding. That's non-repeated protein coding. There's actually a bunch of repeated sequences, but non-unique protein coding, which basically says what makes each of us different from each other, at least in terms of protein coding sequences, is only 1.5%. So it's a very small amount of our genome that actually is different in terms of protein expression. On the other hand, a lot of what's present in bacteria and also in archaea, these are almost completely protein coding sequences. So nice, simple things to look at. Which gets me to model organisms. Why do people not all work with humans or their own particular favorite microbe they happen to have found in the soil, their one particular plant, their animal, etc.? If you want to understand real fundamentals of biology, like molecular biology, like we're talking about in this class, it's really nice to have lots of people working on similar, if not identical, organisms, because there's a whole bunch to do in all of these different organisms. And different organisms turn out to be much better for different experiments than others. Bacteria, and when they say bacteria in this textbook, 90+% of the time they mean E. coli. There's a huge amount of bacterial diversity that has nothing to do with E. coli. Really fascinating stuff. Take microbiology, learn all about it. But as far as the molecular biology, and again, what's true for the E. coli is true for the elephant, very basic aspects of molecular biology. A lot of that has come out of the study of E. coli. Small genome, easy to do genetics, and was just one of the easiest things to work with in the lab. You can grow a whole bunch of E. coli really quickly, and there are no E. coli rights activists out there. Very convenient. In terms of thinking about archaea, another big group of organisms, again, most molecular diversity, as most of you knew about. One of my favorite model organisms for that is Syllabus. Sulfolobus is an extremophile. It grows optimally at 80 degrees Celsius and pH 3, so basically boiling acid. But other than that, it's really easy to work with. Also has a very small genome. This is, in fact, a TM of Sulfolobus right here, and there are a bunch of viruses that infect it, which is the other reason that I'm excited about working with it. There are a number of eukaryotic model organisms that are used. I mentioned yeast already, the smaller eukaryote, not the lower eukaryote. A couple of reasons that people really like to work with yeast is mostly because it's really easy to grow, they're great genetics, and it's got a small genome. So those are the main reasons that people work with Saccharomyces cerevisiae. C. elegans is a little bit bigger. Dr. Suzanne Estes here. How many of you know Dr. Estes? Fabulous professor. Her lab works on C. elegans and related nematodes. C. elegans is also a really wonderful genetic system. Unlike yeast, which is basically single cellular, C. elegans has multiple cells. C. elegans is also transparent, so it's really easy to see all of these cell divisions, and it eats E. coli, so also makes it relatively straightforward to work with. Been extremely well understood from the process of development. And again, also very good genetics are present in that organism. The model plant, you may remember from that last slide, a lot of plants have really ginormous genomes, which makes doing genetics really hard. Turns out a lot of these turn out to be because there's been multiple genome duplications, not just gene duplications, but genome duplications. Many plants are tetraploid, some are octoploid, etc. So huge amounts of DNA, and even in those haploid genomes, lots and lots of DNA. Arabidopsis is relatively small genome, pretty good genetics in Arabidopsis, and it also grows really fast relative to something like a sequoia or an oak tree. So that's why Arabidopsis has turned out to be one of the model organisms if you want to study plants. Drosophila, this was one of the very first genetically tractable organisms ever to be used. Anyone seen The Fly Room? Really cool film about the Columbia development of genetics, actually very early genetics. We'll talk a little bit more about this later on. But just trying to connect genes to functions, genes and genetics, the real origins of genetics were in Drosophila. And it turns out that, that fruit fly you see flying around your compost, certainly around my compost, is really easy to work with. You can grow a whole bunch of them. Again, I know this from my compost. And they also have a number of mutants, and it turns out their genes, and this is one of the kind of surprising things, a lot of the genes in the Drosophila genome turn out to be really similar to genes in our genome, and how they work is also very similar to a bunch of genes in our genome. So we'll talk more about these later on. More recently, the zebrafish, and we've got a whole colony of zebrafish over in SRTC, have become a nice model organism for studying vertebrate development. And they've got a relatively small genome. There's pretty good genetics. They're also easy to follow in a developmental process. Probably the standard organism now, model organism for things like us, are small and furry because A, they have a relatively short lifespan. B, you can grow lots and lots of them. They breed the way you want them to breed, as opposed to the last one on this list, which has a nasty tendency to breed with what it wants to breed with, rather than vice versa. So that's another one of these nice model organisms, and incredibly powerful genetic tools have been set up in studying mice, most musculus in particular. Probably the most impressive of those are these gene knockouts. So you can literally have one gene and knock it out. Even we've got ways of knocking in genes into these mice. And some of you, how many of you have heard about CRISPR? Yeah, a few of you. Well, we may have a guest lecture on CRISPR. One of my graduate students is really into it, so he may come and talk about that as well. CRISPR has actually really revolutionized these modifications of genomes, and now people can do genome modifications in non-model organisms, which is a really, really powerful technique here. And then, humans are a model organism. Why humans as a model organism? Okay, we don't breed the way we're supposed to. We've got big genomes. Why are humans interesting? One, there's a lot of diversity in us. Just look around. There's quite a bunch of diversity which is here, even though our genomes are incredibly similar to each other. The second one, and probably most important, is we self-report. We tell other people that we're sick. We go and talk to doctors. We're interested in sequencing our own genomes, and so we do a lot of that. There are literally billions of people, one species on this planet. So there's a huge amount of data which are available for humans. And one thing to think about, I always mention this in my class, I'll probably mention it a few more times, pretty much everybody in this room is going to have their genome sequenced, which is kind of amazing. I don't know if you've done that already, but that's the whole concept. You will know what your genome sequence is. Do you want your employer to know what your genome sequence is? Do you want your insurance company to know what your genome sequence is? Interesting questions. Yes?
I was just going to ask you, what do you think of the quality of the 23andMe, or like, there's a lot of literature out there that talks about that.
Yeah, so the question is basically, again, I'll try and paraphrase this. What do I think about commercial genome sequencing organizations which are out there? We can talk more about that later. It turns out that most of these don't actually do whole genome sequences. They're very much exon genome sequences, and we'll talk about exomes and things a little bit later on. But yeah, we can certainly talk about that. There's some genetic issues which are way beyond the scope of this course that we could definitely discuss. Probably best to do online or D2L discussions, that kind of thing. There's also a human genetics course, by the way, I think, which is being offered next term. I hope it is. Dr. Duffield usually does that. Pardon? Oh, it's now? Is this term? Okay. Yeah, excellent class. So this is just basically the pictorial way of showing what I just told you about here. Saccharomyces cerevisiae, single-celled eukaryote that undergoes most of the molecular things that you think about eukaryotes doing, with the exception of development and multicellular differentiation. Here's C. elegans, the worm with literally, well, I think it's the hermaphrodite that has 959 cells, and it's known exactly where each of those cells come from, starting from the fertilized egg, which one goes to each of the other ones. Here's our friend Drosophila. This one I think is a wild type. I don't see any obvious mutants on it. White eyes was the very first of those. Here's a Arabidopsis. It's not quite to scale. This is a mature Arabidopsis plant. It's probably about that tall, so they're really quite easy to work with. I didn't mention Xenopus as a model organism. Turns out it's really hard to do genetics with Xenopus, but it's really easy to get eggs. They're huge. And some of the very first cloning experiments were actually done with frogs and with Xenopus. And we'll talk a little bit more about those later on. Zebrafish, again, developmentally, this is a really nice system. You can grow lots and lots of zebrafish and look at their genomes in terms of what's going on there. And then our favorite model organisms, the mouse and the human here. I wanted to put up a scale. This is again from the textbook. On this side, looking at the time of divergence of these various different organisms. And just for comparison, again, remember this is this tiny little twig right here on the big tree, the tree of life, as it were. So the differentiation here, let's see if I can get the pointer over here. Oh, we can use this. Between humans and mice is about 40 million years or so. So molecularly, that's not had much time to change. And if you just look over here at the percent amino acid identity, and this gives you a pretty good idea if it's a homolog, between mice and humans is usually about 85% identical. So extremely similar. As we'll see later on, anytime that you've got an amino acid sequence that's more than about 30% identical, it's going to have an identical structure and usually very similar function. Yeah? So how much details we need to know about the model organisms? And sorry if I paraphrase here, it's going to be on the test to some extent. What kinds of level of detail I expect you to know? The answer is I don't know, but I would go back and look at previous exams. I'm not going to expect you to know that there are 959 cells in a hermaphroditic C. elegans. That there are small numbers of cells and that we know the complete lineage of those cells, yes, I think that's a reasonable thing to ask. But the best thing to do is to, again, look at my old exams and see what I expect. I'm always going to expect that you know the size of the haploid genome is about 3 billion base pairs. That you know that the latest release of the human genome, which I think is 36, is 3,238,325,000. I don't expect you to know that. Does that help? A little bit? Good. Okay, so that being said, this 85% sequence similarity means, again, they're probably going to be extremely similar in terms of their function. And a nice example of that is the genetics right here. It's a little hard to see, but this baby actually has a mutation in exactly the same gene that this mouse has a mutation in. And it leads to this discoloration right here in the middle of the forehead. So this is a really nice example on why using mice is a really good model system for understanding humans. Also really good for understanding mice if you happen to care about mice, and there are lots of reasons to care about mice as well. So that brings us to the end of Chapter 1, all the stuff I was supposed to talk about in the first lecture, and now we're, 3/4 of the way through the second lecture. So syllabus, subject to change. So quick review, and just ask me questions on this as we go along. Central dogma, DNA to RNA to protein. Big difference in central dogma, and somebody asked me a question about this online, also on D2L now. The big surprise that came out after the first central dogma is that RNA could go back to DNA, the reverse transcription. Reverse translation can't work for a number of different reasons, and that might be a question that could come up on an exam. Why does reverse translation not work? We talked a little bit about genomes. We'll talk more about genomes as we move through here. And again, thanks for the question in the back there. We're talking about everything, not just coding sequences, all the non-coding sequences as well, which we understand a lot less about. Gene families, orthologs, paralogs, homologs, and then model organisms. So more questions about this. Yeah? So the question is, are orthologs implicated in speciation or develop after speciation? I will obfuscate on that question and say yes and no. So it's not clear. And the whole question about speciation is a really excellent question, and biologists argue about it all the time. So what that means is, I think, a very open question. Yeah, but as far as we're concerned, again, maybe to back up a little bit, as far as molecular biology is concerned, they're going to be two different species if they're going to be, giving us orthologs. Okay, so let's do all of chemistry in half an hour. Well, all the chemistry that I'm going to expect that you know for this class. This should really be a review. I'm going to go through it really quite quickly. A couple things about atoms, very general chemistry. Then we'll talk a little bit about the weak interactions, and most importantly, water. Without water, there probably wouldn't be life, and that's partly why NASA is looking for water in all kinds of places that are not here, if we're going to try and find life. Everything we know about life says it's dependent on water. Weak interactions, these are the van der Waals, hydrogen bonding, and ionic interactions. This is really what's critical for biology and biological interactions. It's lots of weak interactions in water in particular that means things can change. If things can't change, then we're not alive. We will talk a little bit about some of the strong interactions. Most of the strong interactions we're going to talk about here are going to be covalent bonds, and a little bit about how we go about making covalent bonds. And the main thing about making covalent bonds is, again, with my model down here, to talk about going from monomers to polymers, monomers to macromolecules. And we'll spend a lot of time talking about that for the rest of the class, and we're not going to get to protein structure today. I'm sure we won't. So what atoms do you need to worry about? Periodic table. One of my students actually had the periodic table tattooed on his arm, and he had to cover it for his chemistry exams. But you can ignore most of the periodic table for biology. In fact, you can just concentrate on hydrogen, carbon, nitrogen, and oxygen, and then a little bit of things like phosphorus and sulfur. The rest of that, really, you're not going to care about. So great. Simplified thinking about chemistry for the rest of the class. Chemical bonds, again, we're really going to mostly be talking about two kinds here. Covalent bonds, shared electrons between two atoms. Non-covalent bonds are ionic bonds. You have electron transfer from one nucleus to another nucleus, giving differences in charge. It's going to be these charge-charge interactions that are going to be giving us things. What's a bond energy? Again, hopefully all this is review. If it's not, then we might need to talk about what things you might want to do to follow up on understanding things. At the top here is, again, a log scale. Very often we're going to use a log scale here, looking at various different bond energies. Average thermal motion is down here. Non-covalent interactions, so these are going to be our weak interactions, are just a couple of times more than just the random fluctuations in water. So any single one of these weak bonds is really not going to do very much for you. So you need lots of them. And so the take-home message here, if we're talking about biological interactions and different dynamics which are happening here between different molecules, it's going to be lots of these relatively weak interactions coming together here. By comparison, covalent bonds over here are about 100-fold higher. And so just random thermal motion is not going to break most of these covalent bonds, whereas random thermal motion will actually break or at least compete with a lot of these weaker interactions. What are those weaker interactions? They're van der Waals interactions, hydrogen bonds, and ionic interactions, but these are ionic interactions that have usually been set up by acid-base chemistry. So last time I talked about biochemical polarity or molecular biology polarity. That's saying that one end of this DNA molecule here is different than the other end of the DNA molecule. Chemical polarity has to do with separation of charge. So separation of charge, and the classic one of these polar molecules is water. Slightly negative at one end, slightly positive at the other end. That's because the atoms have different electronegativities. Oxygen really loves electrons. Hydrogen doesn't love them quite as much. And so you end up with different ends here of your molecule. And generally, because water has this differential in charges, those charges are going to like to interact with each other. Water loves to interact with water. Partly why water is liquid over such a very wide range of temperatures, but also in terms of interacting with other chemicals. And in general, a polar molecule that's not water is going to interact very well with water, not surprisingly, because like and like. So those are going to be your hydrophilic molecules. The vast majority of biological molecules, and particularly the ones we're going to talk about in this class, are going to be hydrophilic. And polar molecules, you also have nonpolar and hydrophobic molecules, and we'll talk about those in terms of proteins a little bit later on. So what is a hydrogen quote-unquote bond? Hydrogen bonds really are polar interactions, electrostatic interactions, again having to do with how much atoms like to steal electrons or pull electrons either towards the nucleus or not pull them as close to the nucleus. This is basically, you have a proton in the case of hydrogen, which is between two different electronegative atoms. A couple of important things about this. In three-dimensional space, for these to work, they have to be well aligned with each other. They've got to be lined up. So if you have something which is offset a little bit, that's not going to form a hydrogen bond. You're not going to be able to get that energy from these interactions with each other. And hopefully all of you remember that how the two strands in DNA fit together is really about hydrogen bonding interactions in between, in the middle. So again, relatively weak and directional kinds of interactions. Van der Waals interactions are even weaker. They're extremely weak, about 10 times weaker even than hydrogen bonds, which are, again, 100 times weaker than covalent bonds. And again, questions about numbers here, that kind of thing, 10-fold, 100-fold. Exact numbers, people are going to argue about them. But 10-fold, 100-fold, those kinds of things I think are reasonable. So van der Waals interactions are tenfold less than you have for hydrogen bond interactions, which means to have van der Waals interactions that may make any difference, you've got to have lots and lots and lots of them. And van der Waals interactions are also very dependent on distance. And so if you look here at this curve, van der Waals interactions are only actually attractive at a very small window. If we're looking here at the x-axis is this distance between atoms, and on the y-axis is attraction, or for that matter, actually repulsion. So if things get too close, these are repulsive. If they get too far away, they're extremely weak and basically make no difference. It's only at a very, very defined area where van der Waals interactions actually make a difference. But on those, if you've got enough of them under these if you get a really good hand-in-glove kind of interaction, these can be very strong and very useful in terms of biological interactions. Hydrophobic interactions. What's a hydrophobic interaction? I'm not going to call it a hydrophobic bond. I like to think of hydrophobic interactions as hydrogen bonds between water. So I mentioned polar being hydrophilic, non-polar being hydrophobic. So what's hydrophobic? Doesn't like water, doesn't form polar interactions with water. Okay, so if you have this kind of interaction, what's going to happen is the fact that water forms hydrogen bonds really well with itself is anytime you've got a nonpolar, a hydrophobic molecule in an aqueous solution, the water is going to want to form hydrogen bonds. And the way that the water can maximize the number of hydrogen bonds that it forms with itself is to push those nonpolar molecules together. And if you look at oil and water, oil is a classic example of a hydrophobic molecule. What happens? Forms these globules. So that's exactly what happens in biological systems. You've got nonpolar. Those end up getting basically squished together by the presence of all the polar interactions that interact with water. Water is always present at very high concentrations, and again, it loves to form hydrogen bonds with itself, which is pushing these hydrophobic things toward each other. Let's move on here. So, van der Waals interactions, hydrogen bonding interactions, hydrophobic interactions, all of these things together, when you add them all up, start to come to a relatively high energy, allows you to have interactions between molecules, and these are biological macromolecules, that are specific, which is one thing, because again, if there are hydrogen bonds, they've got to be aligned with each other. Van der Waals interactions, they've got a really close, great fit to have these be at all attractive. So to have these, you end up with a specificity. So you have lots of these individual interactions, but they only work under one or relatively few conditions. So A, you need that, and B, because they're relatively weak, you can have these interactions be transient. So sometimes they're interacting, sometimes they're not interacting with each other. And there's actually quite a nice movie that's connected, I think, to the textbook that you can take a look at, looking at some of these interactions with each other. Here, this side is shown as actually having lots of charge-charge interactions. We'll talk about how you get those in just a second. This one, I think, is actually a slightly better image. This is the one from our textbook. It's in a different textbook, where you've got hydrogen bonds, ionic interactions, and then hydrophobic here in the middle, having all of these different interactions going together with your individual molecule. So how do you get these charge-charge interactions? Charge-charge interactions in biology, sometimes you have ions and ionization, sodium chloride, potassium, etc. Those are the exceptions, at least as far as we're going to be talking about here. Much more important for us are going to be acid-base reactions. So acids losing a proton, bases basically picking up a proton again from water. In that process, what happens is you end up with a charge, because again, you're moving these protons around. So acetic acid is a nice example here, has a proton which is relatively easily lost to water. We talk about nucleic acid. Why is it an acid? Because the phosphates are losing hydrogens to the water. So that's why it's an acidic molecule. DNA, nucleic acid, it's losing that. So what does that mean in terms of a charge? If it's losing a hydrogen, it means what kind of charge? Negative charge. So what's the charge of DNA? Negative. Exactly. So that's all we really care about in terms of acid-base chemistry. We're not going to talk about oxidation, reduction, etc. There's a nice overview in your textbook, but I really hope that you've learned that already. So now switch over and talk a little bit more about our molecules that we'll be discussing for the rest of the class. Again, very few atoms, not very many different kinds of reactions. What molecules are we going to care about? Well, they're sort of basically big molecules and little molecules. The little molecules that we're going to talk about are mostly important for helping reactions to take place, having to do with free energy, but also as precursors for putting together the bigger molecules. Most of the time we're going to be talking about bigger molecules. When I say bigger molecules, I mean proteins, I mean DNA, I mean RNA. And these are all things that are made going from individual building blocks or monomers to polymers. So you think about sugars, go to polysaccharides. What we're going to mostly be interested in is amino acids going to proteins, and nucleotides going to nucleic acids. To do these kinds of things, hopefully, I think I've got this slide in just a second here, you realize that going from monomers to polymers is energetically not easy to do. So you have to have some kind of energy inside the cell, and this is what most of those small molecules are about. And we're really going to mostly concentrate on adenosine triphosphate, our friend right here, with all of these extra phosphates out here on the side. We're not really going to talk about sugars and lipids for the rest of this course, but they're very important in terms of cell biology and other courses a little bit later on. If you just look at a cell, why do we care about most of these things? In a cell, not surprisingly, it's mostly water. We already talked about aqueous chemistry. We're talking about biology. We're talking about biochemistry. Most of a cell is water. And again, why NASA's looking for water as well on other planets. But other than the water, what else are in cells? And it's mostly these macromolecules: proteins, RNA, and DNA. What's most of this RNA in a cell? What's most of the RNA? Ribosome. Why? Because it's making all these proteins. And it turns out that ribosomes are not great catalysts, so you need lots and lots of them. So between what you need to make your proteins and proteins, that's the vast majority of what's not water inside any given cell. How do you make these things? The chemical reactions we're going to be talking about in this class come down to basically two: hydrolysis and condensation. Having to do with what molecule? Hydrolysis is, hydro is water, condensation is water. So it's all about aqueous chemistry. So you have a condensation reaction where you release water. That's bringing two molecules together here, A and B. Hydrolysis, adding water, pulling those two molecules apart. So that's basically all the chemistry we're going to be talking about in this class. Cool. Great. All kinds of really fascinating chemistry, biochemistry, etc., which we're not going to talk about because we've got other things that we want to talk about here. If you think about these different reactions, hydrolysis is a energetically spontaneous reaction. Condensation is not. And why is that? It's because you're pulling two molecules together, you're forming a covalent bond, but also what you're doing is you're lowering the entropy of the situation. So what do we mean by lowering entropy? And actually I've got one slide ahead of myself here. This is one of my favorite slides in the whole textbook. Can you tell I have kids? So the idea here, as far as entropy is concerned, oops, go back here. Had it here. There we go. This is what we see on the right, most of my house, and requires energy to get it over to what's on the left. So, well, so actually this should looking this way around. So to your left, my right, so you go from the disorganized state, the monomers. How do you get to your organized state, to monomers, which is what we care about? This is the life, even though, again, I'm sure my kids would argue that this is life rather than this being life. But as far as we're concerned, this is life. And so we need to go from monomers to polymers. And for doing this, we need energy. And we need a particular input of energy in terms of making each of those individual bonds. So that's that condensation reaction, pulling two things together. How can we do this? Because it's a non-energetically spontaneous reaction. Most biological reactions, again, these polymerization reactions, are going to be going in a positive direction as far as free energy is concerned. That is now a non-spontaneous reaction. How do you do that? Well, the only way you can do it is if you have other reactions that they are connected to. And this is the coupling aspect that we'll spend a little bit more time talking about in the next lecture. But just to finish off, this is yet another way of looking at this process. We've got our food molecules, which get broken down, generate energy, but they also generate heat. And so any of these biological reactions, which are going to be a spontaneous reaction, are going to be generating heat. And if you have a good way of coupling this energy and lost heat into an anabolic pathway. Now, I think about catabolic pathways and anabolic pathways. The way I like to think about catabolic pathways, what do cats like to do? Make things smaller. They're really good at chopping things into smaller pieces, like your skin on your arm and those kinds of things. So that generates the energy and precursors which you need for the rest of your biological reactions. So we'll talk about this other way of getting energy other than eating, i.e. photosynthesis, on Friday. So we'll see you then.